Okay, let's dive right into this explainer. We're going to take a wild ride through the absolute fundamental rules of cellular biology. We'll look at how we actually discovered them and how modern science is uncovering organisms that, honestly, shatter the very rules we thought were absolute.
So we usually think of bacteria as these tiny, invisible little specks, right? Just unseen forces floating around us. But I want you to imagine a microbe so massive, it literally breaks the laws of biology as we know them. A single cell that you could hold in the palm of your hand and actually see without any specialized equipment. It sounds totally like science fiction, but I assure you, it is entirely real.
Meet Candidatus Theomargrida magnifica. It's a newly discovered centimeter-long bacterium, literally roughly the size of a fruit fly. It has this complex life cycle with two distinct forms, and it even compartmentalizes its DNA into membrane-bound organelles. This one single organism throws our entire traditional concept of bacterial size limits completely out the window.
To really put that into perspective, we kind of need to anchor ourselves on a logarithmic scale. Your run-of-the-mill typical bacterium, it sits way down at just 1 micrometer. Even an animal cell is only around 10 to 100 micrometers. So knowing that standard baseline is exactly what makes our fly-sized anomaly such a biological marvel. It's growing orders of magnitude beyond what was thought theoretically possible.
The truly crucial number to keep in mind here is 100 micrometers. That is the absolute hard limit for the human eye. Anything smaller than that, and we absolutely need a microscope to even know it's there. And for centuries, that physical limit kept humanity completely in the dark about the microscopic universe and the very building blocks of life itself. Now, to really appreciate how nature breaks the rules today, we have to look back at the sort of scientific true crime story of how we established these laws in the first place. It all kicked off back in 1665 with Robert Hooke, followed by this slow realization that plants and animals are made of cells until the ultimate breakthrough in 1855. That's when Rudolf Virchow popularized the massive concept that all cells must arise from pre-existing cells.
And wow, this was a massive paradigm shift, because for centuries, humanity was completely stuck on the ultimate locked room mystery. People believed that life just magically popped into existence from non-living matter, a crazy concept known as spontaneous generation. People genuinely believed that if you just left out some rotting wheat, it would literally turn into mice. No joke.
Francesco Redi was the guy who took the first real swing at this myth back in 1668. He used a brilliantly simple setup: open, cork-sealed, and gauze-covered containers of meat. He basically proved that maggots don't just magically appear from rotting meat, they only show up when flies can land and lay eggs. But of course, maggots aren't microbes, so the fierce debate over invisible life just kept raging on.
It actually took the careful methodology and absolute genius of Louis Pasteur in the 1860s to finally close the case on where microscopic life comes from. He did this with his incredibly famous swan neck flask experiment. He boiled nutrient broth in this specialized flask with a long, curved neck, leaving it totally open to the air, but cleverly protected by the geometry of the glass itself.
And this right here is his trap. The elegant swan-like curve of the glass caught the airborne dust and bacteria, completely preventing them from contaminating the sterile broth. But the second you break that neck, the broth quickly rots. Pasteur proved once and for all that the invisible air around us is just teeming with microscopic life, officially killing the idea of spontaneous generation.
This revelation was a total game-changer. It paved the way for pioneers like Ignaz Semmelweis to push the incredibly radical idea of washing your hands, and Joseph Lister to start sterilizing surgical tools. Couple that with Robert Koch establishing strict protocols linking specific microbes to specific diseases, and boom, we birthed the germ theory of disease. So why does all this matter to us today? Well, when we look at modern data of microbial infections in the US, we can clearly see the real-world stakes of germ theory. Bacteria, things like E. coli, staph, and Clostridium, cause the vast majority of fatal pathogenic infections, though we are definitely seeing fungi catching up as emerging pathogens. So understanding these organisms is literally a matter of life and death.
But hey, bacteria are not just invisible little villains. They're actually incredibly diverse architects. They use their specialized cell walls to form a wild variety of shapes just to survive. You've got the spherical coccus, the rod-shaped bacillus, combos like coccobacillus, and even these rigid spirals called spirochetes. Their shape is deeply tied to exactly how they survive in the wild.
And this structural diversity perfectly sets up how we organize life at a grand scale. Take the domain bacteria. They generally have rigid cell walls and are practically everywhere. and contrast them with the domain Archaea. Archaea have completely unique genetics and chemistry, which is super cool because it lets them thrive in some of the absolute most extreme environments on our entire planet.
When we actually map all of this genetic diversity out, we get this stunning, massive tree of life. It beautifully reveals just how vast the eubacteria domain is up at the top, followed by Archaea. And if you look really closely down there at the bottom right, that tiny little isolated branch is us, the eukaryotes. Plants, animals, fungi, we are just a tiny blip in a heavily bacterial world.
Now, if we zoom into one of those bacterial cells, we need to completely shatter a boring textbook myth right now. If you look inside a microbe, you won't find an empty water balloon with a few things casually floating around. Actually, scratch that, it's not even close to a balloon. That idea is just completely wrong.
The reality is that a prokaryotic cell is absolutely stuffed. It's totally jam-packed with thousands of ribosomes churning out proteins and raw, unmembraned DNA just hanging out in the center. The inside is so intensely thick and gelatinous that the cell constantly fights extreme internal pressure just to keep its outer membrane from popping like a tire.
But perhaps the absolute coolest biological rule break of all brings this full circle right back to us. Ancient bacteria didn't just stay separated on their own little branch. According to the endosymbiotic theory, ancient proto-eukaryotes actually swallowed aerobic and photosynthetic bacteria whole. And instead of digesting them, they formed a symbiotic relationship, evolving them into the mitochondria and chloroplasts that are literally powering your body right this second.
So we've seen a massive, centimeter-long bacterium that theoretically shouldn't even exist. We've traced the incredible scientific journey to prove life doesn't just poof into existence from nothing, and we've learned that we are basically walking, talking bacterial power plants. It really makes you wonder, if the so-called laws of biology are this flexible, what other fundamental rules of life are just waiting to be broken out there in the invisible world?